Science Gallery Bengaluru is a new institution for public engagement with research. Psyche is our 5th exhibition season and our 3rd exhibition season, which is completely online. In this season, we are exploring the complexity of the human mind. It's with great pleasure I would like to introduce you to our 7th speaker in the public lecture series, Professor Upinder Bhalla, and his lecture today is titled Memories at the End of the Rainbow. How does the brain work? Professor Bhalla came to this question in a roundabout way. First, through his interest in physics at IIT Kanpur and Cambridge. There he realized that biology was full of amazing mechanisms and the brain especially so. He did his PhD at Caltech in the then new area of computational neuroscience. And throughout his career, he has used computers both as a tool and a metaphor to understand the brain. He has been at NCBS TIFR in Bangalore since 1996, and here he has studied how animals recognize and find odors and how memories are formed and stored. We are very excited to listen to this lecture today evening, but before I hand over to Professor Bhalla, I'd like to invite you all to also sign up for our upcoming programs, the Twin Lives of Psychoanalysis in India, which is a public lecture next weekend by Shruti Kapila, as well as the first in a series of 6 performances, Hamlet's Life, by Preetham Koil Pillai and Chanakya Vas on 15th April at 5 p.m. I'd also like to remind the audience that there will be an interactive Q&A at the end of the lecture, so please do drop your questions in the Q&A box and do fill out the feedback form in the link so that we can know how we can make our programs better. Now I'd like to hand over to Professor Bhalla for the evening. Thank you.
Thank you very much, Madhushri, and thank you to the Science Gallery Psyche team. And thank you to everybody who has come in this evening on a Sunday evening to listen to me talk about my favorite subject, the brain. So I will now try to get my screen to share properly. And should that exercise be successful, we will proceed. Okay, so this is a lovely picture taken by Srikar Krishna of the institute where I work, and it sort of resonates with the topic, memories at the end of the rainbow. And what I'm going to do today is I'm going to give you a glimpse of how you bridge the very deep physics and chemistry and biology of it to the experiences that are so essential to what we call memory. So let me start by expanding a little bit on the metaphor. So this is how a rainbow is formed. You have light coming in through and bouncing off a little droplet of water. And it forms a virtual image at the magical angle 42. And this is well known. René Descartes actually had an analysis of this many, many years ago. And you form a virtual image of the rainbow. So what has this got to do with memory? So one possible answer is that maybe it's because the rainbow and memories are both an illusion. I think that's a little bit of a oversimplification because memory is actually very real for us. And certainly rainbows are very real. In fact, this rainbow has been captured on a camera. So even though it may not be physically there as you can touch it, but certainly it's there physically in the sense that you can capture its light. So let's try another answer. It keeps receding as you approach. Now here we're getting a little bit more metaphorical. You know, as you try to understand the subject a bit more, maybe you find that it's more and more complicated and more wonderful than you expected. And maybe something like that is happening with the rainbow. Okay, fine. But that's not such a good way to analyze it as a scientific topic. You want to have something a little bit more hypothesis-driven, something testable. So this is what I will use as the launching point for this discussion. That memory is real and it has a physical basis, which I will give you some glimpses of. But our brains do something in forming a memory, forming a recollection that shape it into something special that related to all our experiences. So what is memory? Let's start with that. Well, here's a form of memory that you are probably all familiar with. It's a computer hard disk. And how does it store information? Well, it stores information basically through retention. That is, You make physical changes in this thing. Small magnetic zones are altered. And the physics of the material is such that it takes a very, very long time, years, for that memory to decay, for that physical change to decay. And so this is one suggested mechanism, though, as you will see, it's not now the dominant one. It's a suggested mechanism that your brain forms these structures called synapses. And if they were to endure and retain their properties for a very long time, then you would have storage through persistence. That is, any changes that are embedded in these physical structures will remain because the physical structures remain. And the reason why that is not such a great biological statement is because, as it turns out, these synapses, and as we'll see, the molecules inside them are actually very dynamic. They do not last a long time. They turn over on the space of minutes to days. So persistence memory is perhaps not the way we do it. Okay, here's another kind of memory. This is a standard DRAM, and you stick it in your computer and it will store information, but it will only store information while the power is running. And the reason why it is limited in this manner is because it stores information through charge, and the charge over time decays, and it needs to be boosted. It needs to be refreshed in order to retain the information. So it has to be boosted back up before it has decayed down to nothing. And something very similar was proposed many, many, many years ago for how the brain might store information. That is, you could have a little loop of cells which are connected to each other. These are neurons, brain cells. And this neuron connects to that neuron, and that neuron connects to that neuron, and so on. And you have something to store, like a phone number. You set up a reverberation whereby the activity of one cell refreshes the information stored in the next one, and that refreshes that in the next one. It refreshes the circuit to retain the fact that there was some activity present. So this is reverberation, and it is still one of the plausible ideas for how memory might work, but it's not so great for long-term storage, because when you stop thinking about the phone number or when you turn off the computer, the memory goes away. So let me bring up a third kind of memory. And this is something which will be familiar to all of those who have done some kind of engineering or have looked at logic gates in high school. And this is a very simple logic gate circuit with two logic gates. These are NANDs. I won't go into the detail of them. But the property of this memory is that because of the feedback here, so here's input A, input B, here's the two outputs, and the outputs connect back into the input of the other gate. And this property of feedback, this characteristic of feedback, is crucial for this class of memory, which is called bistability. And bistability, I'll get to that in a little bit. The principle of this is that it will store information completely reliably as long as power is presented. As long as the thing is kept, as long as the battery is active, it will store information. And this kind of memory, for example, is present on your CPU, what's called cache memory. It's very, very fast access memory in a computer. And the metaphor that this applies to, as we shall see in a little bit, is, again, feedback processes. The key word is feedback, that occur through, in this case, chemical gates, chemical logic, that is present in every single one of your cells, in fact, in every single small protuberance in your cells called dendritic spines. And so I'm arguing that the same idea of feedback as a way of storing information through bistability is something that might take place in every single cell, in every synapse in your brain. So we'll come to this in more detail in a little bit. But now let's step back. Yeah, at what level should we be looking when we want to understand memory? The brain, I will assert, actually cannot be decomposed into any one level of analysis. If you do so, you throw away the essential parts of how the brain works. For example, just to give you something which might be familiar, you can have molecular changes such as mutations, which will affect everything down this cascade all the way to behavior. So some mutations, as you well know, will affect people's ability to remember. So you have a molecular change at the genetic level, cascading all the way through all these levels, leading to a behavioral change. So the point is then that analysis of brain function, memory in particular, has to take into account all of these levels. It has to be a very multidisciplinary, integrative approach. And this is what we would like to try to take. And what I'll do today is I will have a glimpse of what is happening at the area and circuit level, and I'll also have a glimpse of what's happening at the molecular level, but stuff is happening all the way through. So let's now take a little tour through the different scales, the sizes of what's going on in the brain. So this is the brain, and it's about 15 centimeters or so long, about 1.5 liters of volume. And this is where you are, so to speak. You are here. And compared to this, the size of a neuron is like this little dot over here you see in the corner. So let's zoom in now. Let's zoom in from the centimeter scale to the submillimeter scale. In fact, to be precise, to a 0.5 micron scale. And so this is a computer model made by my good friend Eric de Schutter many years ago of one of the beautiful neurons in the brain called the Purkinje neuron. And this is about 0.5 millimeter from side to side. And this is the next scale that we look at. But now let's zoom in still further. We're going to go down to the size of this little dot over here, which is like a small piece of the cell at which a lot of the action is taking place. So now let's zoom in a bit more. So here is a 5 micron cube of tissue, which was very, very meticulously serial sectioned using electron microscopy. And then even more tediously, it was reconstructed so that they were able to build up the three-dimensional structure from the successive little images of the tissue. It's a very, very challenging task, and now it's in fact the basis. This approach is the basis for what's called connectomics, but we won't go there today. But let's have a look at this. So this yellow thing is one of those dendrites that you saw in the previous slide. It's one of these branches. And now you can see somewhat smaller structures, these little mushroom-shaped things sticking out. And these are called dendritic spines. And on them, you can see these little red dots, which have been, of course, artificially colored in. And these represent the synapses. And that's what I indicate by the size of the synapse. And what the synapse does is it is the connection point from the axons. Here's one axon indicated in green. There are hundreds of axons in this image. We're only showing one for clarity. Yeah. So this axon makes a number of synapses, and these are the connection points between the axon and the dendrite. And this is where a lot of the action takes place that I'll be talking about. This work was done, as I said, many years ago by Kristen Harris, Mary Kennedy, and Terry Sejnowski, and it has been followed by a lot of fantastic work in subsequent years. So now let's zoom in still further. We are going from 5 microns to 0.5 micron. And now we're looking at one synapse, and to be more precise, at the presynaptic terminal, the axon side of one synapse. And now we're in the range where we can sort of see in these little stringy things, we can see the actual molecules, the proteins. Some of them are stringy, some of them are round. There's a whole lot of vesicles, which are little bags of chemicals, of neurotransmitters and other good things. There are these structures, which are composed of proteins. These are microtubules. You can see the cell membrane. You can see a lot of proteins sitting on the membrane. Right down here, you can see the synaptic release zone. This is where the neurotransmitter is released and makes signals propagate to the next cell. So what we've done is we have zoomed all the way from the size of the brain down to the level of individual molecules. And throughout this, I really want to emphasize everything is completely jam-packed with information processing machinery. Think of each of these little strands of protein and vesicles and all as having some computational capacity, and now scale that up from this molecular scale to the size of the whole brain, and you get a sense of just how densely packed is your head full of computational machinery, which is a good thing. You want your brain to be able to do a lot of good things. Okay, so now stepping back from the molecular level, let's go to another definition of memory, or the behavioral definition of memory, which was first nicely systematized by the great Russian scientist Ivan Pavlov, of whom you've all heard, I'm sure. And what he did was he was actually interested in digestion. He was interested in the salivary enzymes and so on. And so he had this experiment going where he had dogs, and he had little ways to measure their rate of salivation. And not surprisingly, but he was a good scientist. He observed something very interesting. So in his lab, not surprisingly, when you gave the animals food, they salivated. This is what's called an unconditioned stimulus. This is something the animals don't need to be trained on. They do it naturally. And he also knew, not surprisingly, that if you just ring a bell or do some other random stimulus, the animal isn't going to be particularly interested in it, right? It's certainly not going to salivate because of it. But because his lab ran to a very organized schedule, what would happen was that every mealtime in the evening, I guess, a bell would ring, which indicated to the staff that now is the time to feed the dogs. And so you had this association, and that's a keyword, the association between the bell, which is originally a neutral stimulus, and the food, which is the unconditioned stimulus, which causes it to salivate. So you have this association many times, and the dog is salivating every time it sees the food. But then something interesting happened, and this is where being a good scientist really matters. He noticed something, it's called serendipity. He noticed something that he wasn't looking for, and he followed it up. What he noticed was that dogs started salivating even before the food came. They started salivating in response to the conditioned stimulus, to the bell. And this is classical conditioning. That is the term given to this form of memory. So fine, so this is something you all know. I've gone through this at some length because I'm now going to take you to a cartoon dog. A cartoon dog, in this case, with just 3 neurons. And the 3 neurons are a food neuron, the bell neuron, and a controlled stimulus, a light neuron. Now, of course, a real dog has a lot more than 3 synapses. That's why I'm calling it a cartoon dog. But let's see how far we can go with this. So here's the food, and the food is connected to the food neuron, activates it somehow, and the food neuron causes salivation to occur. So this is the unconditioned stimulus and the unconditioned response. Food, saliva, via the activity of this cell. Not a big deal. Now, let's say that we have association with this bell. An association as in the bell and the food are simultaneously presented time and time again. And now let's further stipulate one more key point, which is that the bell connects to the food through what is called a synapse. This connection point is a synapse. We're going to spend some time looking at these. So when the bell neuron is active and the food neuron is active, which is happening now because the animal is being trained on this, these two things are active presynaptic and postsynaptic. The food neuron and the bell are active at the same time, repeatedly, time and time again. The synapse is active at the same time as the food neuron. And as we shall see in a little bit, this sets up a whole set of molecular changes which cause this synapse to go from being a weak connection, where the bell would never have originally triggered saliva, through repeated association becomes a strong synapse, a switched on synapse, such that now bell on its own is able to trigger enough activity in this cell to cause salivation. So this is, of course, a complete cartoon version of what's happening. It's far more complicated, but this gives you an idea of how you go from a fairly sophisticated behavior, how you can sort of analyze it in terms of mechanisms, and as we shall see, molecular mechanisms that can relate the change of how synapses function. And just to remind you about the control, the light is also presented as many times as the food is, but it is not associated with the food. Therefore, the activity does not coincide, and therefore this change in synaptic properties does not take place for light. And so light never is able to trigger salivation, only the bell is, and of course the food. Okay, so this is a cartoon dog and a cartoon illustration of how you can get a behavioral-like effect from very simple fundamental molecular cellular properties. And we will dive into this in more detail. But let me just, again, step back a little bit, right? We've been talking about Pavlovian conditioning, a very, very simple, very, very classic kind of learning. Humans do much more than that. And I'll just give you one example from a lovely little book called Mind of a Mnemonist by, again, a Russian neuropsychiatrist, Alexander Luria. And he had this patient, an unfortunate man, you would be surprised, because this poor fellow, his patient had a perfect memory. And the problem, as you shall see in this little example, was that the memory actually, this perfect memory actually debilitated him. It made him hard for him to function as in normal society. Let me give you an example. So Luria was in the habit of presenting his patient with some table of numbers, a lot more numbers than I have here. Here I have only 16 numbers. He presented him with a table of numbers and asked him to look at it and memorize it. And then a year, 5 years, 10 years, even 15 years later, he would ask him to recall what was in that table. And this gentleman always did so. He did so through something called synesthesia, but I won't go down that route. That in itself is a very interesting topic where there's some kinds of cross-wiring where, for example, certain numbers are associated with certain sensations like color or taste or smell. Be that as it may, here's one example of a table which I thought was particularly interesting. And this is something which none of you will have any trouble seeing as a pattern. Yeah, so you have a 1, 2, 3, 4 in the first row. Then you have 2, 3, 4, 5, 3, 4, 5, 6, and so on. So this is a very organized table. And if you and I see this, we will immediately spot that it's organized, and we remember not the numbers, but we remember the organization. But this gentleman didn't. And this is the key to his problems in life, that he looked at this thing, and sure enough, he recalled it. He recalled it 10 years later. But it was only when it was pointed out to him that these numbers formed a pattern that he said, oh, I didn't see that. So he actually went and memorized every single number in every single location without seeing that it formed a pattern. And forming a pattern is extracting information, extracting the structure of things is an essential part of how we think and also how we remember. And so, I also want to remind you that this is so far, so many levels of complexity far away from this simple 3-neuron dog. But underneath it, as I'll try to explain, we still have the understanding that is happening through synaptic changes. So let's move on. So here's another glimpse again from history. This is a classic study on this, again, unfortunate man by the name of Henry Molaison, a different person. He suffered from intractable epilepsy. And as a result, Scoville and Milner, well, Scoville was a surgeon, they had to perform a fairly drastic surgery, which would not be done nowadays because we know the effects are really debilitating, in which they removed the locus of the epilepsy, the hippocampus. So this is the brain after. And fine, it cured the epilepsy. But what it did was it completely ablated his ability to lay down new memories and to have short-term memory. For example, the doctor who had done the surgery had to introduce himself every morning. And yet Henry Molaison, who, he liked baseball, he could reel off the statistics of games that had happened before he had had his surgery. He could remember what the president was and what the president said, but he could never anchor himself in the present day after his surgery because he had no short-term memory. And it gets even more interesting there that he, as I said, his long-term, his memories that were formed before his surgery, he retained. Those long-terms were retained. He could not lay down new memories, but he was still able to learn skills. For example, in one thing, he was taught to draw things in a mirror image, which is a skill that you and I can learn. He was also able to learn this, but he didn't know that he had learned the skill until he was presented with the task and he did it. And there are other very interesting things about him. For example, one day a scientist came in to test whether other kinds of memory were affected, and he had a pin in his hand, and he shook Henry Molaison's hand, and Henry Molaison, of course, said, ouch, and was not very happy. So then this gentleman walks out again and comes back 10 minutes later, at which point Henry has forgotten everything. He walks in and he tries to shake his hand, and though they greet each other, the patient refuses to shake his hand. So some kinds of memory are affected by this, some are not. And as I said, the brain is way, way, way more complicated than the simple circuit I gave you. These are just some glimpses into it. So as I said, a lot of the action happens in this part of the brain called the hippocampus. This is what it looks like in your head. The red banana-shaped objects are your hippocampi, or I should say hippocampuses, the two lobes of your hippocampus. And it's not such a huge region of your brain, but it's extremely, extremely important. Here's an example of what it can do in a system called place cells in mice and rats. And in fact, this work won the scientists, the Mosers and John O'Keefe, the Nobel Prize a few years ago. And what this beautiful work shows is that the animals form a very specific kind of memory called place representation. In other words, you recognize where you were, and you can actually look at what individual cells are doing. Let's take this particular case. So here's the activity of a given cell, which is indicated in red when it is active, as the animal is running around in a certain arena, in a certain box, say 2 meters on a side. It's quite a large arena. The animal runs around at random. Those are the little black tracks. And the cell is active only in this region. And this is kind of a heat map of that activity. And you can do the same thing on a linear track, which is illustrated here. And basically, the region that the animal is familiar with is tiled, is fully filled up with cells that are active in one place or another. And so by looking at the activity of these cells, you can figure out where the animal is. And it remembers that. So the animal can go somewhere else. The cells will be active in some different way, different context, maybe not active. You bring it back into the same arena, the same room, and they will be active in exactly the same positions as they were earlier. So this is another important kind of memory. So this is memories of place, and place cells is the name given to this kind of activity. We'll talk about a different kind in a moment. But now let me change gears a little bit. Here's a marvelous quote from one of the fathers of neuroscience, Santiago Ramon y Cajal. He called neurons butterflies of the soul, the beating of whose wings may someday, who knows, clarify the secret of mental life. So what I'm going to do now is I'm going to dive into a little bit of what is happening at the level of neuronal activity, first in networks and then subcellular. So where does one look for memory? And as I've stressed, one cannot look at it in exclusion at any one level. One has to look across scales. And I'm going to look at it at sort of the scale somewhere between here and here. And then I'm going to look at it at this scale down here. I'm going to look at it at the systems level first. And as I said, memory is happening at all of these levels, at the systems level, the area level, the circuit level, synapse level, molecular level. These are all part of memory. So don't get me wrong. I'm going to focus on a couple of things, but that's just because of limited time and the limited number of things that I could do to look at it. The field as a whole has thousands of people looking at across all of these levels. So this is a kind of experiment that we do in the lab. So we work on mice, and what we do is we implant little windows, and I'll explain why in a moment, little glass windows over their brain, such that it lies over the hippocampus, which I already told you about. So that's the mouse hippocampus. Yeah. And we make a small hole in the skull, and we put the glass window on it, and we cover the whole thing with a nice solid metal plate so that the animal can be placed under a microscope. And this, in this example, this is an old slide. This is done with what's called a fluorescent dye. Nowadays, we do this with transgenic animals, which you don't have to inject any dye at all. The animal is genetically modified so that it has this fluorescence. And this is a very special kind of fluorescence. It is a bright signal. The cells become bright when they are active. In other words, if the cell is quiet, it is dim, it is not very bright. And if the cell is active, if it's firing a lot of action potentials, it becomes bright. So let me show you what that looks like. First, let me illustrate to you how we look at these things. So this is one of the custom-made microscopes that we have built to do these projects. This is the objective. This is where the animal sits. I'll show you a bit more of that in a moment. Somewhere in the dim distance is a very, very powerful laser, which provides light to all of this. And here's an image that comes from recording from this instrument. So we train the animals because, of course, we're interested in memory. So we use something extremely similar to what Ivan Pavlov used. We have a sound. That's the speaker there. Remember the bell? But for the unconditioned stimulus, we're not giving the animal food. What we're doing is we're giving a puff of air to the eye. And what happens when you have a puff of air to your eye? You blink. And that's what this animal does. And so exactly the same as Ivan Pavlov had, we trained the animal by having an association between the puff of air and the light. So here's the light going on, then about half a second later, the puff of air happens, and the animal learns eventually to associate the two kinds of stimuli. In other words, it starts to blink, I'll show you in a moment, before the puff of air comes. So here then is one of the very first, that's why it's so grainy, one of the very first images we got from the microscope, where the 2-photon microscope. And I'm still completely excited by this because here we are literally watching the activity of the cells, which are these little bright spots blinking on and off. And their blinking on and off is literally them responding as we watch to different stimuli that are coming into the brain. Yeah, it's just so marvelous. Now, this is only a few hundred cells, 100 cells or so. Nowadays, this technique is able to record from tens of thousands of cells. Some variants of this can go even more than that. It's quite amazing how technology has enabled us to literally see what's happening in the brain. Okay, so we would like to know many things. Here's a set of questions that we've been working on and will continue to work on. What is the life cycle of memory in the hippocampus? You have to form it. What is happening to the cell activity when the cells start to say that I am responding, say, to this place where the animal is running around? How do they represent it? In this case, we use a slightly different thing where it's not representing place, but representing time. What happens when one learns something new? How does one manage to remember many things at the same time? And what happens when we forget? So these are just some of the questions we ask. I won't go into the details of this. Instead, I will move forward and give you fast forward to one of our earlier papers where we looked at formation of a memory. And so just as before, this is the light stimulus. Here is the sound stimulus. Here's the air puff stimulus. And this is the activity of 6 different cells. As I said, we recorded from hundreds of cells. The people who recorded, I should stress, were not me. They were Mehrab Modi over here and Ashish Dhawale, who is now a faculty very close to us at Indian Institute of Science. Mehrab is finishing up his postdoc. So these very talented grad students, they recorded activity, and what they found was that over many trials, so there are 3 trials represented in each of these traces, initially the cells were not doing very much. They were sort of randomly active, yeah? So the little asterisks indicate what were the peaks of activity in this small period, in this small time window. But after learning, something really remarkable happened, which is that the cells started to become reliably active at a certain time. And in fact, not at the same time, they became active at slightly different times in the period between the conditioned stimulus and the unconditioned stimulus, between the sound and the air puff. So in some ways, they were doing in time what you saw earlier was being done in place. Here, instead of representing locations, they're representing times following the stimulus. And so they're sort of making a mental map of the association and the different times that are present in the association from beginning to end. And so this has been well studied. Previously, it had been shown at a very different time scale. And these are therefore called time cells. That is, they anchor the animal, we think, in time, just the same way as place cells provide a reference in space. So now let me change gears. So we were looking at activity of whole cells just a little while ago. The cells we were looking at, by the way, are pyramidal neurons, which look like this, which are also very beautiful neurons that Cajal classified. So now what we're going to do is we're going to zoom in and look at very, very small structures at the molecular level. So that's, as I said, we're going to go from the cell level right down to the molecular level. And let me do that. So as I stressed already, this is incredibly jam-packed information processing machinery. And let's get a glimpse of what it can do. And of course, what I'm going to present is a very small subset of what it's actually doing. It's very, very complicated. We're very far from fully understanding all these things. So this was a model I made now many years ago, 20 plus years ago, to study some of these interactions. And it's already pretty complicated, even back then. What we can do now is we can look at the the properties of these interactions at the level of these individual synapses. Let's zoom in again a little bit more, which are each of these little button-like, mushroom-like things. These are the dendritic spines. We can analyze the chemistry and the voltage activity happening at all of them at the same time in these computer models. We can ask what happens when input is coming in a certain sequence, which is what this particular study was about. We're simulating activity coming on all 5,000 or so of these throughout the cell body. We're modeling all the chemical reactions that are taking place. And what we find is that this is selective to a particular sequence of inputs, and that when the same inputs are activated in a random order over here, it produces very little activity compared to that. So activity is indicated in red. So this is just an illustration of how now we can go from what's happening at the single synapse or the single spine level to what's happening at the whole cell. But I'm going to take you back down to the single synapse, in fact, down to the molecular level. If you remember, right at the start of the talk, I told you a bit about something called bistability. This is the property of many systems, physical, biological, chemical, of being able to retain in two states. For example, light switch is bistable. You can click it on and you can click it off. It stores information in the sense that once you clicked it on, it'll stay on. Once you clicked it off, it'll stay off. So let's now take this to a slightly more abstract form. If you think of this as a kind of energy profile, you can think of it literally as the shape of a track in space. So you could have, and here's a marble. So you could have a marble sitting in one low state, state one, and it's stable there in the sense that you can push it left or right, but it will always roll back to the middle point. So that is stability. Now, if you push it hard enough, you can push it over the divide, over the bump, and this is called an unstable state. There's various math, there's a whole bunch of maths behind this. And it can go into the second state. And once it's in the second state, it'll be stable over there. So that's why it's called bistable. It's got two stable states, each of which is resistant to perturbation, but a strong enough stimulus will cause it to switch one way or another. Okay, so what does this have to do with chemistry? So let's think of this as a little molecular circuit, right? So we have a molecule A, let's say, connected to a molecule B. Let's say it's some kind of enzymatic step that causes A, so such that A will activate B. And because it's a circuit, because it's a loop, we say that B will activate A. And you could imagine that in the resting state, A is off, B is off, so nobody is activating anybody and everything stays off. Now let's give this system a strong, my earphones have fallen off. A strong stimulus, and now A becomes active. It turns on B, B is active, therefore it turns on A, and this is a stable state. It is one where the system will remain intact, continuing its activity, as long as the cell is alive, and we'll see that in a little bit. If you zoom down to single molecules of A and B, it's like there's a pool of A's, the triangles, and B's, which are the octagons, and they feed back on each other such that if they're in the inactive state, they stay there. If they're in the active state, they stay there. Okay, so now let's take this to the cell. What we'll do is we'll take it to a small piece of the cell, and these little structures which I told you about called dendritic spines, I've drawn them in a very schematic way over here. So here are A's and B's, and at present they're in the active state. But now something happens, which happens, of course, as I mentioned, all the time, which is that the molecules get degraded. They turn over. So that's what's indicated by these crosses. And so eventually you don't have those molecules in the spine anymore, in the dendritic spine anymore. And you can clearly see that this would cause a rundown of memory. It would just dissipate. Of course, the cell is alive, so that doesn't happen. You get new molecules synthesized, but the problem is that these new molecules, so to speak, they don't know what was the original state. They have no memory because they're new. But this is where the bistable element kicks in. Because this set of molecules, the dark blue ones, are in the active state or in the inactive state, whichever it is, they are the vast majority, and the system is stable in that state, whichever it may be. And so these new molecules are just like a small perturbation in the overall activity. And so they become recruited to be part of the same activity. They become entrained to be part of the feedback process, and they also take up the activity. And this is not too different, if you think about it, from how oral traditions were passed on for many generations. That is, you have the individuals will come and go, they will die, and new people will come along, new students will come along, and they imbibe the traditions, and then in due course, they too are part of the process that sustains them. So this is how you can store information at the molecular level through the formation of switches. And this takes us all the way back to the very early slides in the talk, when I said that, something happens due to the association. And similar molecular logic occurs that ensures that the activity of these kinds of switches is triggered when you have association between the food neuron and the bell neuron repeated many, many times. It causes the feedback loops, which may not be the kinds that I've shown you. There are many candidates. It causes these feedback loops to kick in, and the thing switches, and the synapse gets switched into a highly active state, into a more transmittive state, such that the bell is now able to trigger saliva. And so we've gone all the way from a molecular view of how memory is formed to the circuit level, the cellular level, and from there to the behavior level. And it's happening, as I said, through all of the interplay at many, many levels of how the brain functions. Okay, so I'm going to now start to change gears and wrap up a little bit. And what I'll do is I'm going to talk about something which is a hot topic for this day and age, which is artificial neural networks, otherwise known as AI. And what I'm going to try to do is to draw a little parallel here between what the brain is doing and what these AI things do. First, limitations, right? The ANNs, artificial neural networks, AIs, they're very unrealistic. They are not a faithful representation of how the brain works. Yet, they're still very instructive. We learn a huge amount by seeing how these things work, and vice versa. These things, of course, they're called artificial neural networks because they took some of the inspiration from how the brain works. They're very useful. Whether or not you like the utility of being tracked and analyzed all the time is your choice. But certainly they can do amazing things, such as you will see, do some health analysis, figure out what your face looks like, other things which may be more useful as well. Yeah. And the fundamental operation they do is classification. And here too, the crucial thing is that they have something equivalent to synapses, and this is where information is stored. And one of the key things that they do, which is similar to how humans do it, how brains do it, is that they learn by example. So let me just illustrate what this looks like. Here is the mathematical form that describes how these neurons, not really neurons, these units in an artificial neural network work. They have many inputs. Each input has a weight, and that is summed and gives an output. And weight is the crucial part. This is just like the synapse. This is the part that has a certain strength, which is changed when a memory is formed. And the same fundamental thing happens in a neural network. The same fundamental thing happens in our brains. The strength of connections change when you learn something. Well, you can put many of these things together. You can make multiple layers. In fact, what we call deep networks in AI today is simply many, many, many layers of this kind of neurons, each connected to the next layer and so on. And just to reiterate, the connections between these little arrowheads, these are the quote unquote synapses, and this is where the memory is. What can you do with this? Here's an example. You can, there's several examples of this kind of work. So this one is by Google. They looked at the pattern of retinal blood vessels in your eye, and they had correlative data about various other health parameters. They trained it on a bunch of patients, But the weird thing is that now by looking at the retinal diagram, the retinal image, they could do crazy things. They could predict cardiovascular risk. They could predict age. They could predict gender almost perfectly. They could tell whether the person was smoking. They could figure out the levels of various hemoglobins, the blood pressure, and whether or not the person had had a heart attack. Yeah, all by looking at the eye. So these are subtle things that are embedded in the information which can be learned. Again, memory. And it's happening through synapses, even though they're very artificial. Here's another one that you may have seen. The real faces are these ones, the 5 on top. Here are sort of target faces. I won't go into the details, but the neural network is asked to generate a face based on the real ones, and using this as a sort of a motivator, as a template. And it generates all of these faces. All of these are fake. All of these are artificial faces. Yeah. So these are just examples of what you can do, and it's all down to those synapses. Yeah. This, of course, is done in monstrous servers, machines, data centers. Here's just one of them, and it is gigantic. And remember, you can do it all in your head. So just to summarize, AI is a very inefficient way of using huge amounts of energy and memory and training to solve these problems, but it's still very limited. Humans are very good at doing this. They're very efficient, but they can be beaten by AI now in such games as chess and Go. And so far, we are able to do it more efficiently, but, well, the future is bright, or otherwise, as you may wish to think. So let me summarize. Yeah, we've looked at many scales of activity from the whole brain right down to the molecular level. We've gotten a glimpse of how cellular and synaptic memory can lead to things what we observe as a scientist as changed behavior. But so in this sense, memory is real. It has a physical basis. We can measure these things. But the experiential part of it, the part that that we associate emotions and context with, that we draw inspiration from, that comes from a huge amount of other stuff that's going on in the brain. I'll give you a glimpse of what I mean through this beautiful line by Tennyson. And this kind of links the rainbows, and which is an arch, to the topic that we've been going through. All experience is an arch, wherethrough gleams that untraveled world whose margin fades forever and forever when I move. And so it's a beautiful picture. It's a beautiful piece of poetry. But this is I think this is the point at which memories go from being, just bits and pieces, bits of synapses being active and so on, to being much more than that. The essence of it is that all experience is part of this. That is, everything you have learned, everything you remember, is associated in different subtle ways when your brain is active. And all of these things contribute to how you remember something, and how, in remembering it, you also recreate the experience that led to that memory. So yes, we can get a sense of what's happening at the physical level, but just the same as we can with rainbows. But the perception, the impression it makes on us is not simply a playback. It is a combination of all the experiences, all of the context that we have learned as humans over our entire life, which may relate to that memory. And so that is, in that sense, I think that this is a great way to think about how we can combine a physical understanding of what's going on in changes to the sensations that lead to memory. So let me just put this in another context. This is my mother. So she was the one who introduced me to poetry and this particular thing. She was, among many other things, she was a teacher, an academic. She was also an avid gardener, as you can see here. And she spoke these words to a group of students, as I am speaking to you, I think to try to give you a sense of, the wonder of and the incredible preciousness of as many experiences you can get to be part of your life. And for that, she is my mother. She was my mother, and always grateful to her. So with that, I'll end. I'll say thank you to all of you for listening. These are my students, my lab at present. Many people have funded me over the years. And thank you to Science Gallery and Psyche, and to all of you. That's it. Thank you.
Thank you so much, Professor Bhalla, for the lecture. It was really quite fascinating. I think I want to almost go into your lab and see some of these experiments happen in person. But before we get, I just like to also invite the audience to share their questions in the Q&A box, and we can ask Professor Bhalla to answer them in a few moments. And I think while people pull together the thoughts and type the questions down, maybe I could start off with a question or two. So one thing that I sort of noticed earlier on when you were talking about the different scales, all the way going from genes and molecules to, of course, then behaviors. I was wondering, a lot of it was about what innately affects, right? Like our genes, which affect memory formation. What is the role of environment? So does things that happen to us as we grow up or as we develop, or in the case of even in a mouse, would this affect the ability to form memories? And I mean, I also come at it from the sort of very social perspective, where now we're sort of bombarded with excessive amounts of information, especially early on in life. So would this at all change how memory formation might occur?
Right. Now, that's a great question, and it is one that I think exercises a lot of people. Let's start with the first part of it, which is, does environment matter? Yes, it matters enormously. Yeah. Genes matter to some extent. We know that. If you have a mutation, you will have certain limits in how your brain can function. But environment also sets enormous limits on how your brain can function. For example, if you do not learn speech by a certain age, it's very, very hard to learn it later. If you aren't exposed to light up to a certain period, your visual system doesn't learn. I mean, there's a lot of learning even in something you think innate, like vision, right? So development is extremely dependent on stimuli, and which is why we encourage, and it's biologically, for example, we know that rats raised in an enriched environment are healthier, they're smarter, they're more active, they're more capable. And the same is true for humans. So a richness of experience is something we should all strive for. And I think that sort of came across in the poetry as well. Yeah, it is part of what makes you human, to have a wide variety of experiences. It's part of what makes your ability to appreciate things stronger. Now, in terms of overstimulation, well, that's an interesting question. I don't have an answer to that. I think the jury is still out. I think the brain is extremely resilient. We can learn to handle many things. I mean, think about it, right? We did not evolve to read. Yeah, we did not evolve. Likewise, we certainly didn't evolve to sit in front of little screens for a large fraction of our waking lives. But I think we can handle these things. For that matter, we did not evolve to learn to do math. So these are all things that we can learn. The brain can adjust. It's going to be a learning process for all of you to how to balance your lives to continue to get the richness of experience that you need to become full people, and that includes richness beyond your screen. It's the whole world. There's a whole world out there.
So there's already a question in from one of our audience members. So I'll start with the first one. What about false memories? Do these follow the same kind of memory formation process or patterns, or is there some different phenomenon happening there?
That's an interesting question. So there's varieties of false memories. I won't get into this. It's not something I'm expert in. But you're absolutely right. Just like you can form plastic changes, you can modify your synapses through real experiences. You can modify them, for example, through dreams. You can modify them also through just thinking about something. And the question isn't whether you can modify them. The question is, do you have in place the mechanisms, the other mental resources to tell which was real and which was not? Yeah. And that is something which is actually, I think, really important in this modern age with information overload. You have to learn to discriminate between what's fact and what isn't. Yeah. Fake news and real news. This is part of your repertoire. This is part of what we've had to do as a species throughout. And you can tell between dreams and reality usually. Similarly, you can usually tell between false and real memories, but there are medical conditions and other situations where you fail to do this. They are all based on the plasticity, but it's all the context that makes you tell the difference.
Wow. There's another question that's quite interesting. So this is from Samyukta, and she first thanks you for the lecture, and then she goes on to ask, does the Pavlovian conditioning work the other way? So would showing food to the conditioned dog activate parts of the brain that hear the sound of the bell ringing?
Oh, that's a lovely question. You should come and do that experiment. I don't know. Okay, I don't know if this experiment has been done. It seems likely because people have analyzed these things over the years, but I don't know the details. It's a very plausible hypothesis because connections in the brain go all directions. Yeah, the hippocampus is particularly notable in being a place where all kinds of sensory and other contextual information comes. So I would not be surprised if what you're hypothesizing is true, but I do not have an answer.
Okay. That's an interesting experiment. I think Samyukta, you can definitely see if you can try that one out. I think we can take one last question. This is from Ankita. Her question is related to dreams. So why do some of our dreams remain retained in our memory, but most are not? And how does our sleep, for instance, affect memory formation?
Okay, no, that's again very interesting question. Not my area, but I will tell you what I know about it. When you sleep, your brain is in a very different chemical state, if you like, or the neuromodulators are in a very different state of functioning. Yeah? Obviously, you're asleep, right? So these are usually at a level where they are not very conducive to storing information, right? The kinds of things that are conducive to storing information are really strong emotions. So you'll remember something vividly if it's associated with some, very significant event in your life, some strong emotion. Dreams are sort of at the other end of the scale. So your sleep definitely affects the level of these neuromodulators, and that affects your ability to remember these things. Also, because to a fairly large extent, dreams are garbage, your filtering system does a good job of saying, okay, this doesn't really hang together. And so you lose it because it isn't really a coherent thing. But dreams do affect your behavior. They affect a lot of things. Dreaming is essential. There's a lot of work that suggests that at some level, the process of dreaming is important in reinforcing patterns of activity that are salient, that are worth retaining. And this is also part of formation of memory, but anyway, it's a marvelous question and a lovely field to think about.
Okay, so I think this is a great point for us to wrap up the session. Thank you so much, Professor, for answering these questions so interestingly, and thank you so much to the audience for coming up with some interesting thoughts at the end of this session. I just want to remind everyone that the recording of all our lectures are available on our YouTube channel. So in case you missed out any of our previous events, do check it out. And if you really enjoyed the talk today, there's also a walkthrough of Professor Bhalla's lab by his student Anzal, titled How Neurons Learn, a live lab session. So you'll actually be able to see that amazing rig that we saw today in Professor Bhalla's talk and see how these experiments happen in real time. And also, if you enjoyed this talk, do consider watching Professor Ravi Mudashetty's lecture in Kannada, which was Tracing the Trail from Memory to Neurons, which actually talked about something similar as well. I'd also like to encourage everyone to please fill the feedback form and to register for the next week's programs, and to definitely visit the exhibition website while you're here. So thank you, everyone. Have a lovely Sunday night ahead and see you next weekend. Thank you so much, Professor Bhalla.
Thank you all. Have a good weekend. Well, what's left of it.